Hello, everyone. Good morning, good afternoon, and good evening. Mark Estigarribia here again, Managing Director from MSQ Ventures. We are honored to host Dr. Sean McCarthy, CEO and Chairman of CytomX, to our MSQ CEO Webinar Series. Today, we are excited to discuss cutting-edge therapeutics for the treatment of cancer, specifically learning more from Dr. Sean McCarthy from CytomX's leading position in the growing field of conditionally activated localized biologics, powered by its ProBody technology platform. CytomX's robust and differentiated pipeline comprises therapeutic candidates across multiple treatment modalities, including ADCs, T-cell engaging bispecific antibodies, and immune modulators such as cytokines and checkpoint inhibitors. CytomX has also established a strategic collaborations with multiple leaders in oncology, including Amgen, Astellas, Bristol Myers Squibb, Regeneron, and Moderna. Sean, how are you today?
I'm doing great, Mark.
Excellent. Where are you calling from?
So I'm here at CytomX headquarters in South San Francisco, California, the birthplace of biotechnology.
Great. Well, MSQ is based in New York City, and our audience is global in nature, spanning 20 countries with a strong presence in China. Good evening, Shanghai. I guess, Sean, what we can do is, starting with your fascinating background, please share with us your historic pathway from Chief Business Officer to CEO and Chairman at CytomX, spanning over 12 plus years.
Well, yeah, thanks again, Mark. It's really a pleasure to be here and to be part of this really great series that you guys put on. Yeah, this is my 13th year at CytomX, sometimes a little hard to believe. I joined the company in the very embryonic early days, as I like to say, seven people and $7 million in the bank back in at the very end of 2010. I joined very shortly after the first venture round, which was led by Third Rock Ventures. And I, much earlier in my career, had worked at Millennium Pharmaceuticals, where the founders of Third Rock came from. And so that was one of the connections that led me to CytomX. And immediately prior to CytomX, I'd actually been in the venture field myself at a firm called Pappas Ventures. So transitioning into CytomX, initially my role was, as you mentioned, Chief Business Officer, charged with really helping to think through how to take this very innovative concept, as it was at the time. It really was kind of an idea for some of the early IP filings. The foundational filings had been made, but it was really an idea around the ProBody, and shape it into a company. And I was very kind of humbled after about 8 months to be asked to be the CEO, and obviously it was an opportunity I couldn't miss, and really was delighted to take on the role, and have been running the company now for about 12 years. So it's been incredible fun. We've made a ton of progress. We've taken that early embryonic company to the public markets. We went public in 2015, and we'll talk more about the strategy, the pipeline, and our vision and objectives over the next hour.
Great, thank you. I guess going back a little more pre-CytomX, we noticed your transactional business development you mentioned, and your strategic partnership experience already started with major VCs, Pappas, and big pharma companies. Can you please share a little bit more in terms of your inspirational story?
Well, thank you. I'm a cancer biologist by training, so as my colleagues here will tell you, still very interested in the science at all levels. I trained in the UK with the Imperial Cancer Research Fund, and did a postdoc out here on the West Coast at the DNAX Research Institute, which became part of Schering-Plough, but then became part of Merck. But I already mentioned that time I spent at Millennium Pharmaceuticals very early in my career. That was my first actual real biotech job, a scientist in the lab. And this was back in the mid 90s. And the company Millennium was one of the foremost genomics platform companies. And what, as employees, what we would hear about very frequently was this deal with this pharma company, that deal with that pharma company. And I just got really interested in deal making and seeing the impact that these pharma collaborations could have on the building of a company from the ground up. And I was very fortunate to be able to work with several great mentors there at Millennium and learn about the process of business development. So to fast forward to when I joined CytomX, it was pretty clear to me and to my colleagues that given the novelty of the platform, the innovation inherent in the ProBody therapeutic platform, that partnering was going to be a vital component of our business strategy. And we set out initially to generate enough preclinical data to then go talk to partners about ways to work with them to help us fund the company, build the pipeline, further develop technology, and scale the organization. And that really was the origin of this partnering strategy. So it's been very deliberate and we've been successful. I mean, we, as you mentioned, we currently have 5 major partners, and across the partnerships, we've been able to generate more than $500 million of what we call non-dilutive capital for the company. And this has helped us just really scale the science, scale the pipeline, and also build a very formidable intellectual property base for the platform.
Right. I mean, a very inspirational journey there. I'm sure there are a lot of people listening to your story, and everyone wants to get a piece of your secret sauce to success. Any advice you can share in terms of these major partnerships? I think maybe you can also elaborate on the most recent ones with Moderna and Regeneron. Well,
again, in terms of advice, I think one of the key things for us as a company is to develop a vision early on. And we did that again more than a decade ago. Our vision statement for CytomX is transforming lives with safer, more effective therapies. So we've always been highly focused on the patient, and that's what we're here to do. This is what brings us to work every day, which is to use our technology platform to make the biggest difference we possibly can. And that's reflected in our mission statement, which is to urgently advance our ProBody therapeutic platform for the benefit of patients. And that sense of urgency is critical as well. And then the last thing I would say is, of course, it's all about the team. Just we have a absolutely incredible team, great company culture. We work hard at building, maintaining, fostering a fun culture at the company. We think that you should have fun while you're working because we spend so much of our time working. And we like to say that we enjoy working together, and we celebrate our success together. But we all stay highly focused on our vision and our mission.
Yeah. Well, you're obviously doing something right with all the successes. Congratulations. So I guess now, and thank you for sharing that. Looking forward to now the knowledge sharing of your company's novel approach to transcending the limits of the current cancer treatments. We'll take questions from the audience on the back of your presentation. So Dr. McCarthy, can you please share your screen and start your company discussion?
Yes, happy to. Just give me a moment and we'll hope the technology cooperates here.
Perfect.
Okay. How does that look?
It's great.
Great. Well, once again, Mark, thanks to you and the MSQ team for having us on today. It's a real pleasure to be here to share our story, discuss our platform technology, the pipeline, the partnerships, and as I just mentioned, our long-term vision for the company. We've always been highly focused at CytomX on building this company for the long term. Our goal is to build a multi-product, ultimately commercial stage company focused principally on oncology. And we're doing that with the ProBody platform that we have been developing now for more than a decade. And the fundamental principle of the approach is to localize biologic therapies into disease tissue. And now we're 95% focused on oncology. I'll have a couple of words to say a bit later on about non-oncology applications of our technology, which we're actually quite excited about. Could present new partnering opportunities. But we're all about the field of localized biologic therapies. And we've really pioneered this field. I think there's no question about that. When we got going more than 10 years ago, this was a really, in many ways, an idea that was ahead of its time. And we were the only company really doing this for a very long time in terms of protease-based localized therapeutics. We have now seen a number of other entrants into the field, and we've really, we believe, created an entirely new field of therapeutic antibodies. In a way, we feel that we've reinvented therapeutic antibodies. I will make certain forward-looking statements in the context of this presentation. I refer you to our safe harbor statements in our regulatory filings. We are, of course, a publicly traded company, CTMX, on NASDAQ. So the basic premise of the ProBody platform is to take advantage of what we call cancer's Achilles heel, which is in the process of tumor cell transformation, migration, proliferation, and metastasis. Proteases play a central role in the development and dissemination of tumors. Proteases are, of course, enzymes that process large proteins into smaller pieces. They are, in a way, we refer to them as molecular scissors. And those molecular scissors are very active in proteases, essentially enabling cancer cells to move elsewhere in the body. And it's ultimately, of course, cancer metastasis that results in the most devastating impact of tumors. So upregulated protease biology is an activity, is a hallmark of cancer, and has been known for many decades. There are thousands of publications on this in the scientific literature. So we realized some time ago that we could, if we could engineer therapeutic antibodies such that they were what we call masked, that we could decrease binding in normal tissues to target, and by having the mask engineered in such a way that it can be cleaved off by proteases, so by these molecular scissors can cut the mask off in tumor tissue by incorporating protease cleavage sites, we could achieve more binding in the tumor, less binding in normal tissues, and in doing so, open therapeutic window to broaden the therapeutic window where there's room for improvement, or to create a therapeutic window where actually there was no window in the first place. This has become an incredibly important concept in cancer R&D today, because across so many antibody formats, we all agree in the field, not just at CytomX, but in the field in general, that we can do a lot better in terms of either improving or creating therapeutic window. And that's because, of course, when you administer a therapeutic antibody or any biologic to a patient, it distributes throughout the entire patient. And if it binds target in normal tissues, it can cause damage and destruction. And as we've, in the field, have moved, again, over the last 10, 15 years, we all agree we need better efficacy of cancer therapies. We need deeper responses. We need more durable responses as monotherapies and in combination. So we're making cancer therapies more potent all the time, driving for potency. And in that drive for potency, more often than not, what comes with it is increased toxicity, because we're engaging target in normal tissues in one way or another. So T-cell engagers, antibody drug conjugates, immunotherapies, cytokines, checkpoint inhibitors, all of these innovations in cancer R&D are, in their own way, running into challenges and separating potency from toxicity. So ProBody therapeutics that we've innovated at CytomX are designed to localize anti-cancer efficacy and decrease systemic toxicities. So this is a really big idea. And over the last decade, because of the amount of funding we've been able to bring into the company, both through the partnerships and also having brought the company to the public market in 2015, as I mentioned earlier, we've been able to go really deep on the science in two different dimensions. First of all, we've expanded the application of masking, protease-dependent masking, the ProBody platform. We've expanded it across multiple antibody formats. And so we have programs in T-cell engagers, ADCs, immunotherapies, and cytokines. And we've treated more than 500 patients with different formats with ProBody therapeutics. So we've really learned an enormous amount, and we've got by a long way the broadest clinical experience and the broadest platform across all of these modalities. The second dimension that we've really gone deep in, which won't surprise you, is the protease biology itself. So we've invested heavily in the company in really interrogating the detailed molecular biology and pharmacology of proteases in tumor tissue to enable us to optimize these protease cleavable linkers. And this really forms one of the key pillars of our intellectual property strategy over the last 10 years, where we've built a formidable intellectual property estate around protease biology, protease substrates, applicable across these multiple antibody formats. So that's the scientific foundation. We believe we've built a very strong company as well around the technology. We'll talk more about the partnerships. It's been a very important part of our business model. Will continue to be. We believe we have plenty of room to do additional partnering. We have great partners: BMS, Amgen, Astellas, Regeneron, Moderna. Really terrific partners. We are a great partner. Our team, we really invest in our partnerships. We have robust alliance management. We build great relationships, and we've been highly productive in our partnerships to date. So, and we look forward to doing more in the future. We've been able to build a strong balance sheet. We have pro forma, as of our last quarterly update, Q2, of about $210 million on the balance sheet, which is a great place to be in this challenging market. And we've built a very strong pipeline. The pipeline encompasses today more than 15 active programs across the company. These range from clinical stage antibody drug conjugates and T-cell engagers to a broad preclinical pipeline comprising CytomX wholly owned assets and also a wide range of active programs with our partners. And so if you think about the long term, the next 2, 3, 5, 10 years for our company, we've got incredible opportunities to make a really big difference for people with cancer, just given the scope and the scale of everything that we're doing with the platform and with the pipeline. So what I'd like to do now is move to a more a deeper dive on the pipeline and some of the specific areas of R&D at the company, which I hope will highlight, will bring to life the potential of the technology, the power of the technology, and the promise for what we can do for people with cancer. And I'll start with our work in ProBody ADCs. So, of course, antibody drug conjugates have really come of age over the last 10 years. I mean, really, in the time that we've been building CytomX, we've seen incredible advances now with more than 10 ADCs approved for commercial use. And the concept, of course, is is very well understood. We conjugate an antibody to a cytotoxic payload, and that payload renders ADCs highly potent. However, one of the challenges that comes along with that is that if an ADC encounters target in normal tissue, it can be destructive, it can cause significant damage. There are also challenges with ADCs where the payload itself can lead to certain toxicities, particularly if the linker through which the payload is linked, is conjugated to the antibody, is unstable, can lead to payload release in normal tissues, and this can also contribute to toxicity for ADCs. And so across the field, we've really been trying to work out the rules of ADCs over the last few years and have made a lot of progress. But our view at CytomX from the early days was there are many very highly expressed tumor antigens that would be great targets for ADCs, because of course the ADC has to bind to the target and be internalized to deliver the payload to the cell. There are many highly expressed tumor antigens that have not been pursued as ADC targets because they're all on normal tissues. They're often on normal tissue. So we realized that by masking the ADC, we could open up an entirely new universe of ADC targets. And that continues to be the basis of our program today. Now, I want to say here that what we don't do at CytomX is we don't mask the payload itself. So what we're typically observing in the clinic is, as others will, is typically the case that ADC toxicities and maximum tolerated doses are driven by payload toxicity. And that's true of our programs. What we're doing is we're opening a window for novel first-in-class targets. And hopefully that will become clear as I show some data over the next few slides. So I'm going to talk about two ADCs that we've advanced into the clinic. One targets a protein called CD71, the other targets a protein called EpCAM. So beginning with CD71, and specifically CX-2029, which is a masked, conditionally activated ADC targeting CD71 with a MMAE payload, so the auristatin MMAE. Let's talk about the target for a couple of minutes. So CD71 is a very interesting protein. It's the transferrin receptor. It's been known to be highly expressed on tumor tissue. You can see here, very highly expressed on lung, head and neck, esophageal, certain hematologic malignancies. CD71 plays a central role in iron metabolism. It's the transferrin receptor. And so any cell that needs iron has CD71. And of course, tumor cells, as they're rapidly metabolizing, they upregulate CD71 because they need more iron. Because of CD71's central role in iron metabolism, however, it's also highly expressed on normal tissues, and in particular in areas of high iron need, like the bone marrow. So we realized, though, given the internalization properties of CD71, it's one of the fastest internalizing proteins that we know of on the cell surface. We thought it'd be a great target to explore with ProBody technology to see if we could open a therapeutic window. So we made this DAR2 MMAE masked ADC we call CX-2029. And we've made a lot of progress with this program. So I'll share some very high-level clinical results from our phase 2 expansion cohorts. Our phase 1 dose escalation, 45 patients. We showed an MTD of, we had escalated to 5 mg per kg, an MTD of 3 milligrams per kilogram Q3 week, and clinical activity in several tumor types, which led us to open several expansion cohorts, which we ran in squamous non-small cell lung, head and neck, and esophageal, gastroesophageal junction cancers. And we were very pleased in the phase 2 expansions to see robust clinical activity of CX-2029. The most recent clinical data that we've shared was earlier this year, is shown here, which is a very encouraging 21% confirmed overall response rate and 57% disease control rate in patients with late-stage squamous esophageal cancer. This is a devastating disease in the third line or later setting. These patients have very poor prognosis, and you can see deep and durable responses to CX-2029 in this very difficult to treat tumor type. We also saw clinical activity in several other tumor types. You can see here, this is the squamous esophageal I just showed you. Now we're looking at duration of response. You can see activity also in squamous non-small cell lung and squamous head and neck cancer, less so in adenoesophageal, which is interesting. So CX-2029 has demonstrated clear single agent activity in several solid tumors, including some very difficult to treat tumors like esophageal cancer. And a couple of other points to make about this data. These are unselected patient populations, and so we're not selecting for target. And as we move forward, there could be ways to further optimize response rates by selecting for CD71 expression or amplification. The second point to make is with regard to the esophageal cancer patients, the majority of these patients had received prior checkpoint inhibitor therapy, and the responders, for the most part, were in patients who had progressed on prior PD-1 or checkpoint. So this opens up very interesting opportunities for future exploration and development of CX-2029. In terms of the safety profile and tolerability, CX-2029 was generally well tolerated. The principal adverse event that we saw, which we think relates somewhat to the biology of CD71, was anemia, which we saw in most patients. However, it's a very predictable adverse event, which is in our clinical work to date has been manageable through a range of interventions, including dose reductions, dose delays, and transfusions. In general, patients who responded were maintained on drug for a significant period of time through this strategy of anemia mitigation. So we believe we have a very exciting drug candidate here. We are the first company to open a therapeutic window for CD71, the transferrin receptor, the first company to show clear evidence of single agent anti-tumor activity with an ADC targeting CD71. And we're excited to now consider our opportunities to move this program forward. We had partnered this program previously with AbbVie. AbbVie took a strategic decision earlier this year to not further advance into formal phase 2 studies. It was a strategic portfolio decision for AbbVie. We have actually, we just announced yesterday that we have renegotiated and regained full global rights to CX-2029, and we do see potential partnering of this asset, repartnering of the asset as a potential strategy moving forward that we're considering. So very excited about our work, not just for the potential of this asset for patients with very difficult to treat tumors, but also as a very important proof of concept for our technology platform. So let me move now to our newest ADC, which we're super excited about, which builds on all of the work that we've done in the clinic with CX-2029 and others to really understand even better the rules behind masking ADCs and ProBody ADCs. And so this next program is called CX-2051. This is a wholly owned asset. It's, again, a conditional ADC targeting EpCAM, epithelial cell adhesion molecule. So the configuration of the molecule, this is a program we feel we've really optimized based on everything we've learned in the past that has a higher probability of technical success compared to other ADCs that we've put in the clinic in the past. Why is that? Two main reasons. One, the target EpCAM already has a level of validation. We know that anti-EpCAM drug conjugates have demonstrated clinical activity in patients, but only when they're administered systemically. Why is that? Well, because EpCAM is present on all epithelial normal tissues, and people have tried to develop anti-EpCAM antibodies in the past, but they've been too toxic when systemically administered. However, EpCAM can shrink tumors when you engage it with an ADC. We know that, if delivered locally. So the target has the potential to deliver tumor responses. We've also used a different payload for this ADC. The payload is a camptothecin derivative. So it's a topoisomerase 1 inhibitor in the TCAN class. We have a drug antibody ratio of 8. And the camptothecin is selected to be optimally matched to tumor types where EpCAM is expressed, including, but not limited to, but including colorectal cancer. So a really very important next generation ADC for CytomX. Just a few more words on EpCAM, the target itself. As I mentioned, several companies have tried to drug EpCAM in the past with systemic antibody therapies, but they have not been successful due to toxicity based on normal tissue expression of EpCAM. Like I said, there are good examples, very solid examples of systemic administration of EpCAM-directed therapy causing significant tumor responses. And so if we can find a way to dose an ADC systemically and safely, we have incredible potential to treat a wide range of different tumors. And EpCAM really is expressed on many tumor types: lung, triple negative, breast cancer, ovarian, and very, very high on CRC. So we're super excited to move this program to the clinic. We have a very robust preclinical package showing potent activity across multiple tumor types, including showing here 2 different CRC colorectal models. We have activity across multiple other tumor types as well. And with regard to the preclinical safety profile, we have great tolerability in cynomolgus monkeys. And we're in the process right now of wrapping up our IND-enabling studies. Our GMP manufacturing is going very well. Our GLP toxicology is complete, and we're now adding the finishing touches to our IND filing, which we plan to make in Q4. So 2051 is a great example of tailoring the target and the payload to maximize therapeutic index against this high potential target, EpCAM. And our IND is anticipated for Q4 with clinical initiation in early 2024. And again, a wholly owned, unpartnered program that we look at this asset as having the clinical potential of a drug like Enhertu or Trodelvy, similar payload, broadly expressed target on multiple tumors. And we really can't wait to see what this can do for patients. So now let me switch to another key application of our technology platform, which is T-cell engaging bispecifics. Obviously, enormous potential for T-cell engagers. It's a field that has shown great activity in hematologic tumors. We have several bispecifics now approved in hematologic. We have CD19, CD3 approved, of course. That's the Amgen micromet drug, Blincyto. We've been working for some time on a very innovative strategy here, again, using our masking ProBody platform to open a therapeutic window for a very high potential, but previously challenging target combination of EGFR and CD3. I'll also touch on very briefly on our work with the Astellas and Regeneron. So the concept here, of course, is that although T-cell engagers have made great progress in hematologic malignancies, they've yet to really break through in solid tumors. There's incredible potential, though, to use T-cell engagers to redirect non-MHC mediated T-cell killing to tumors if we can open a therapeutic window. So what have the limitations been so far? Well, there are 2 main limitations to moving. There are many, but there are 2 main limitations that we've encountered in the field in moving T-cell engagers to solid tumors. One is these agents are incredibly potent. So rather than dosing in the typical milligram per kilogram mode that we're all used to for therapeutic antibodies, T-cell engagers are typically dosed in the microgram per kilogram range because they're highly potent, because, of course, they're recruiting T-cells to kill tumor cells. What that means is if the target is present on normal tissues, you can see pretty significant T-cell mediated destruction of tissue in a target-dependent way. So target-dependent toxicity has been a real limitation. And then, of course, there's the general phenomenon, which is still not that well understood in the solid tumor context of cytokine release syndrome, where administering T-cell engagers can lead to broad-based activation of the immune system, high levels of cytokine spiking, leading to significant deleterious impact on the patient. So we are exploring how masking of T-cell engagers can open a therapeutic window by decreasing on-target tox and decreasing CRS. So CX-904 is our lead program. This is our EGFR CD3 bispecific. EGFR, of course, I won't say much about. Everyone knows EGFR is a one of the most validated tumor targets that we know of. It's a protein that drives oncogenic tumor proliferation, and has been a very successful target for many drugs, including antibodies like cetuximab and panitumumab, for many small molecules. But no one's broken through with a bispecific against EGFR. It's got tremendous potential, though, because EGFR is present on most solid tumors. The problem is it's on many normal epithelial structures, and we're all aware of the typical side effect for EGFR therapy with biologics, which is very severe skin rash. So we have optimized preclinically our EGFR CD3. We're masking both the CD3 and the EGFR binding arms. We've shown that we have increased maximum tolerated dose, extended the PK, maintained potent anti-tumor activity, opened a broad therapeutic window based on our preclinical work, a window that we believe is about 60-fold or maybe even higher, broader than the unmasked T-cell engager. And we've moved this program into the clinic. So we're in phase 1 dose escalation with CX-904. We are making great progress here. We are in, as I said, phase 1A. Right now, very focused on characterizing safety and characterizing dose levels that we will move forward into. Our goal is to move forward into backfill cohorts in EGFR positive tumors by the end of this year. So we're on track to initiate those backfills to begin to explore anti-tumor efficacy at a dose or potentially more than one doses that are shown to be well tolerated in our phase 1 dose escalation. So excited about the program, about the potential of our technology. This program is partnered with Amgen. CytomX is running the clinical study through phase 1B. The goal next year would be to sit with Amgen, discuss the phase 1A data, discuss the backfill data, and develop our strategy for moving into phase 1B, which CytomX will run. After phase 1B, it becomes Amgen's responsibility to run late-stage development and ultimately to launch the drug. CytomX retains significant US commercial rights to this asset, excuse me, as and when it moves forward. So great progress, and I hope this illustrates again, the potential and the power and the breadth of our technology in T-cell engagers. That breadth of our technology and the potential has been recognized by not just Amgen, one of our 3 partners in T-cell engagers, but also by Astellas, with whom we have a broad multi-target T-cell engager collaboration. We've made great progress here. We earned a clinical candidate milestone earlier this year for the first candidate selected, and we're working on multiple programs with them. We also retain certain US commercial rights in this deal as well. Again, always keeping our eye on the long-term value creation for the company. And more recently, we were absolutely delighted to bring Regeneron on as a partner as well. So working on multiple bispecific immunotherapies with Regeneron. And I think that with this deal, relatively recently signed, I think it really continues to show and emphasize the depth of the science at CytomX, which we really see as unmatched in the field of conditional activation and protease biology. So I'll wrap up now just with a few comments on some other applications of the technology, just a couple of minutes on immunotherapies. I think we all agree that in the field, we've got more work to do to find the next big breakthrough in cancer immunotherapy. And we at CytomX, we see that coming from a couple of potential areas. One is the field of cytokines, where we're employing masking strategies to broaden therapeutic window for these very potent immunotherapies that are of broad interest across the industry at the moment in terms of trying to localize the tumor tissue. We've focused on interferon alpha, which we think has been a little bit overlooked, actually, but has enormous potential. And I'll also just touch briefly on our work with Bristol Myers. So our interferon alpha program, interferon alpha 2B, actually was the first immunotherapy to be approved in 1986. Actually was approved about a decade before IL-2. And it is known to have single-agent anti-cancer activity against multiple tumors, certain hematologic cancers, and melanoma, renal, and others besides. A very potent cytokine. It has very significant systemic side effects. This is the challenge. So we have made a conditionally active version of interferon alpha 2B. And one of the reasons we really are excited about interferon alpha is it has a dual mechanism of action in terms of its anti-tumor activity. First of all, it plays a central role in antigen presentation, and so presenting tumor antigens to T cells, because of course interferons were first discovered in the context of their antigen presentation role for viral proteins to the immune system. Well, interferons play a critical role in presenting tumor antigens to T cells, but interferons also have direct anti-tumor activity. So we have taken interferon and we have pursued a masking strategy, a dual masking strategy to open therapeutic window. And this is very important data from a presentation we gave last year at SITC, showing that our dually masked interferon retains potent anti-cancer activity. But if you look intratumorally, you can see activation of the drug by tumor proteases activating the interferon so that we see immune cell modulation in the tumor, but we do not see immune cell modulation in the periphery. So the masking is blocking interferon's activity in the periphery, but maintaining it by being unmasked in the tumor. So you see the consistent theme now across our programs, the ADCs, the T-cell engagers, where we are able to open therapeutic window and broaden the potential of these highly potent immunotherapies and now move towards the clinic. We believe CX-801 has a best-in-class profile. We are in IND-enabling studies. Again, GMP manufacturing has gone very well. GLP toxicology has gone well. We're, again, writing the IND. Our goal is to file the IND in Q4 with clinical initiation in the first half of 2024. And again, this is a wholly owned program as well. Just wrapping up on our work with Bristol Myers, really to emphasize the long-standing nature of this partnership. We first entered into this arrangement with this deal with BMS in 2014, in fact, before we were a public company. And we continue to work with them on multiple programs, including CTLA-4. We're very excited about the work and the investment that BMS continues to do to broaden the therapeutic window for CTLA-4 therapy. And the lead program here is what we call BMS 986288. It's a masked version of a non-fucosylated version of ipilimumab, designed to be a more potent version of anti-CTLA-4 therapy, which BMS has in phase 2. Last but not least, on the partnering front, our most recent partnership is with Moderna. This is really exciting because it moves us into the mRNA space. So we're now working with Moderna in mRNA-encoded biologics, and not just in oncology, but also outside of oncology as well. And I think this partnership illustrates a couple of important things. First of all, again, the depth and breadth of our science, but also that there are many ways, many creative ways CytomX can continue to pursue and prosecute our partnering strategy as we continue to build the company. So really excited to be working with Moderna and all of our partners and making great progress in all respects. So to wrap up, it's been a busy time for CytomX, very busy year of execution. We expect some great milestones next year. We are planning to present initial data for CX-904, the T-cell engager, targeting EGFR and CD3 in the first half of next year. We're getting ready to file the 2 new INDs for 2051 and 801. CX-2029, we now have the asset back from AbbVie, which allows us to consider our strategies moving forward, potentially including a new partnership for the program, really in particular following up on that really exciting signal in squamous esophageal cancer. And continued progress with BMS and across all of our partnerships and pipelines. So I know I ran a little long, but thank you for your time. And I hope that was a helpful overview of what we're up to at the company.
Thank you, Sean. I really appreciate that presentation. Congratulations on all your achievements to date. Wish you luck going forward. If maybe you can stop sharing your screen and we can take a few questions from the audience. Give me a second while I gather a couple of these. While you were talking, we started gathering. Here's one. We know one of the strengths of conditional ADCs is it widens the target space for ADCs. How does ProBody Therapeutics manage to do that?
Yeah, this is one of the foundational strategies that we've pursued, and that I just, as I discussed in the presentation, we believe that technology has the potential, as we've demonstrated with CX-2029, to allow us to target highly expressed tumor antigens, even if they're on normal tissue. And CD71 is a really good example of this. And EpCAM, as we're beginning to get ready to file the IND, is another really good example. So again to reemphasize the concept that by masking the ADC, we can bias the activity towards tumor tissue and away from normal tissue, allowing us to open a window for these really exciting new targets. And if there's one thing that the ADC field needs moving forward, in our opinion, it's new targets. And so fundamentally, the ProBody therapeutic platform can open new target space for ADCs by leveraging this strategy.
Great. I think we kind of went through the multiple modalities where ProBody therapeutics has been validated. I guess just a little bit more of elaboration here. So it's been applied to various modalities such as antibodies, ADCs, T-cell bispecifics, and cytokines. Could you elaborate on the potential benefits and unique advantages of using this technology in each of these modalities? I think you kind of mentioned it a little bit, but I think sort of just elaboration here.
Yeah, well, each of these modalities has its own unique features and its own subtleties in terms of the potential for the treatment of cancer and the way in which we leverage the technology platform. So ADCs, as we just mentioned, our goal is to open target space. In the context of the T-cell engagers, we really see our technology as addressing the fundamental need in T-cell engagers today, which is how do we open a therapeutic window at all for solid tumors? I mean, we're really just beginning to see companies make progress here. Our partner Amgen, as an example, has recently released pretty exciting data on their DLL3 CD3 T-cell engager in small cell lung cancer. This is one of the first demonstrations of the utility of T-cell engagers in solid tumors, and to really break through, and that's a pretty restricted tumor antigen. To really break through, we need to open therapeutic window. This is exactly what we're looking to do with EGFR and CD3. Is what we're looking to do with our partners Amgen, Regeneron, and Astellas. And fundamentally, our technology allows us to, again, by masking strategies, decrease binding in normal tissues, maintain binding in tumor tissue, maintain tumor activity in the context of decreased toxicities, such as on-target toxicities and cytokine release syndrome. So we really see ourselves as, right in the mix here of taking on this principal challenge of T-cell engagers today. That's why our partners are working with us, and we see a ton of potential moving forward for this format with our existing partners, our existing programs, and potentially with new partners in the future.
Great. Couple of more. What are CytomX's long-term visions for ProBody technology, and how do you foresee its impact on the healthcare industry in the coming years?
Well, as I mentioned, our goal from the very early days has been and continues to be to build a long-term, multi-product, commercial stage oncology company. And we're well on our way there. We've got a very robust technology platform, a very robust pipeline, strong partnerships, well-funded. And the passion that we have at this company, which is embodied in our vision, is to make more effective but safer, more effective therapies. Because at the end of the day, to really drive the levels of tumor regression that can get more patients into long-term durable remission, we need more active drugs. We need more active drugs that can be combined with each other safely and effectively. And so we see our technology as central to the collective efforts of the industry in making safer, more effective cancer therapies. And of course, if we can do that, when we do that, we will, we'll have made the impact that we all come to work every day to make.
Great. And we have time for two more questions. And a collaboration seems to be a significant part of your strategy, multiple partnerships with big pharma companies. How does this collaborative approach contribute to the advancement of ProBody technology and its potential applications?
Yeah, the partnering has been absolutely central to CytomX from the time we started the company, and will continue to be important. We're not done yet. We're open for business, and we look forward to bringing additional partners on board, whether they're on the platform side, whether they're in oncology, potentially outside of oncology, or whether they're on our specific programs, like, for example, CX-2029. So, we emphasize partnering. We like to partner. We're a very effective and very good partner as well. And our ability to continue to develop the technology, go really deep in the science, while of course protecting our intellectual property and building our intellectual property, it has been and will remain central to us maintaining our leadership position in the field, but most importantly, to maintaining and ultimately realizing our vision to make the biggest difference we can for patients.
One more. So with regard to your global regional expansion goals, I think we've been kind of pressing on that as well. Someone asks, please share, your priorities on your next steps, next 6 to 9 months, 12 months, and are you open to enter the China market?
Yeah, so right now we're based here in South San Francisco. We're a US-based company, and obviously, ultimately, our goal would be to pursue commercial activities on a global basis. We do a lot of our clinical work, of course, ex-US. We work extensively in Europe. We do work in Asia with several of our programs, so we have experience. And we're very open to partnerships on a global basis. With regard to the 2029 program, as an example, it's not lost on us that one of the areas that we have observed we believe some of the most important clinical activity is in squamous esophageal cancer. It's not lost on us that this is a cancer type that is highly prevalent in Asia, and where there is significant unmet medical need. And in fact, a good proportion of the clinical work that I've shared today on CX-2029, the CD71 ADC, has been conducted in Asia. And so, as we now have rights to this program back from AbbVie, we will be considering our strategy, and it could very well involve more clinical work in Asia, in that patient population, where we think the drug can make a real difference, and could potentially involve future partnering, for example, in some kind of a regional relationship. So yeah, we're absolutely open for business on a global basis. Well,
thank you very much, Dr. Sean McCarthy, CEO and Chairman of CytomX. Really enjoyed your discussion, learning more about you, learning more about your journey and your company and your vision. Congratulations again on all your achievements to date. Looking forward to more great news coming from your company.
Well, thanks again, Mark, to you and the MSQ team for having us on. It's been a real pleasure, and look forward to following up.
Great. Thank you so much. That concludes
Take care.
the
Bye-bye.
webinar. Thank you.